can, yes, usually we can start, I think. So
Uh, shall I start?
yes, we can start, I think. We have providing the recording, so others will look on to that. Okay, we can start.
Yes.
We can start.
Okay. Okay. Uh, just a second. Yeah. I hope good evening, everyone. So I hope like everyone was able to download the raw data and finish with pre-processing and then able to do trimming and alignment. Can you please respond that until what have you been able to do? Uh, Hello. Can everyone please let me know till what step were you able to finish? Hello.
Please respond so that we can continue the session. That will be better.
I guess no one is online. I'm not sure. Uh, any one person who can respond up till what have you finished with the analysis? Hello.
What happened? No one is responding. No one has done anything.
In
So, what to do?
I'm not sure. Is they are they even listening? Is my doubt. I'm not
Mm.
sure. Usually, some message response. Okay.
Yes, yes, yes.
Okay. So thank you for the response. Jerry, okay, indexing, Miss Priyanka, understood, install all apps, okay. Anyone else who has done something with alignment also, who were able to manage with alignment? So I hope you are understanding how do you access the raw data from publicly available database and in this workshop. Yes, Mr. Jerry. Indexing usually for every alignment tool takes a long time, which is why I had given a break for yesterday. Indexing, alignment, and variant calling. These three are very computer intensive steps, and you need to minimum give it a time of one entire day. If you have like supercomputers and if you're working in servers, you need not worry. You can give multiple threads and finish it off. But if in your personal system, usually we don't have 16 GB, more than that memory. So it takes long time for us. So until now, like we got a rough idea how do we download the data, how do we index the reference genome, and how do we align our raw data with the reference genome. One important point here is trimming. So you always have to check your raw data with fast QC tool and multi QC tool, and then only take a judgment whether you have to trim low quality reads or not. Mostly you do trim at least one read if in the forward and in the reverse end. That's just to give you an optimal result. But if if your result raw data shows more than 5 reads are contaminated or are low quality reads, then you have to accordingly adjust the filters. One advice of me will be whenever you install a tool in case of any bioinformatics analysis, you need to know all the options. You need to know your data which you are working with, and you have to take a call based on that. So every data is different. So it cannot be a generalized analysis process until and unless it's your own. You have standardized the step and you don't want to change it. So moving further, after alignment, what we'll have to do is we will get BAM and SAM files. I'll just type here SAM and BAM files. We'll also get we can either get a SAM file or we can either get a BAM file. The easiest way to output an alignment is to print a BAM file. I hope you all can make a note of this. Just note down just BAM file. One second. Yes, Dr. Ahmed. Uh, you can switch on your mic and ask your query.
Hello.
Yes, hi, Dr. Ahmed.
Yeah, hi, how are you?
I'm fine. How are you
Um,
doing?
I'm good, thank you so much. So I'm just, is there is any screenshot that you showing because it's not seen? I mean,
No, I for now I have not shared any screen. I'm just saying it out.
okay,
Yes.
okay,
So I
okay,
would like if you could, notice certain points like SAM files, BAM files, and get familiar with these file formats. So basically, every alignment after you finish an alignment process, it will output two files, SAM files and BAM files. But we can only choose to output one single file, that is the BAM file. BAM file is a binary file which the computer only can process. It's not human
uh,
readable at all.
okay, okay, okay. Yeah. So go ahead. I will just be down and listening to what you are saying.
Sure, sure,
Thank you so much.
sure. Uh, Yes, so after you finish your alignment, the main part comes is variant calling. As I had mentioned, one second, I can show you example of a SAM file. I cannot open in my system because it is a very huge file, but I'll share a screen. You can just refer. So when you give your BWA command, that is uh, this is the BW alignment command which I have underlined. So here what I am doing is first I have downloaded the reference genome from GEO database. I had as I showed you last lecture. So after I download it, I need to index my reference genome using the command index. So this is because I'm using a BWA tool. I'll use BWA and then I'll index the reference genome. In my system, the reference genome name is as what I have underlined, and then I can give it any name after indexing, like for example, human genome, or I can just leave it as it is. It will automatically take the original name and it will create index files. So I can show you how the index files look. Uh, yes. Can you see these? The first GCF file .amb.ann.bwd and PAC and SA. So it has created these five indexed files, after which we have to align. Always remember, no alignment is possible without indexing a reference genome. It will never take it. So basically, indexing is the tool does it for its own information and its own coordinate sorting it does. And after that, we perform alignment using this command. BWA is used for most of the alignment processes. So here minus T8 is I have given 8 threads, and then I have given the index name here. If you can see the indexed reference genome name, I have mentioned the index name here, and then I have given my files, trimmed files, which I obtained from Tribomatic tool, and then I've piped it out. Piping means you are inputting this first command's output directly to the second command. This is the second command. I'm sorry if this feels a little out of track because this is a little of a computer language, but this is the simplest way I can explain. After you align, the output file actually comes as instead of this, I can maybe write and show to you. But what is the actual output file is you can either mention as minus o alignment or align dot sam or minus s aligned dot sam. or you can directly give it as arrow and aligned.sam. Either of the three, it will create a SAM file. And then always after SAM file, we have to convert it, we have to sort and convert to BAM file. Because SAM files, even though they are human readable SAM files, we cannot use it for further processes because it's very huge in data size. So what we do, we compress it and we convert it into binary files, binary alignment mapping, BAM stands for, and then it compresses the size also. So this helps us to easily process it. So I give the command for converting SAM file to BAM file is, we use SAM tools. You might have to install them as well. SAM file to BAM file is this command. So what we do is, instead of typing two different commands separately, there is a simple way where we just give two commands with a pipe in between. Like, this is the first command, and I'm inputting this first command's result directly into the second command, that is SAM tools BAM tool sort. It is directly sorting the output of this first command, and it is directly creating a BAM file. So this becomes very easy and less time consuming for us. So you can also, when you're learning Linux in detail, you'll also come across piping, which is a very easy and quick method to finish two processes at the same time. Then comes, these are my commands which I will share after the session finishes today. Then once we have the aligned files, I'll have to do the variant calling. Now basically variant calling means I have aligned all the files, all my sample files together with the reference genome. I have obtained a file which will show me all the genomic coordinates and which samples have mapped to the reference genome with what coordinates and etc. This is all a binary file which we cannot read and understand, but this is what the tool does. Using that tool, we will call the variants. So basically, variant calling can be done with either GATK haplotype caller, which is the mostly used preferred tool in industry and in the research field as well. Otherwise, you can also use Freebase. Now, important thing here is, even the GATK is very highly used, it has to have a very high computer space, very intrinsic computer memory, RAM data, everything. So if you can install, I would encourage you to install GATK. Even Freebase is also fine. There are very minute differences between Freebase and GATK. So you can always decide which one you want to use for your study and proceed. This is a standard command for GATK, where because even this is a Java file, we give it a normal assign a normal memory to the Java file. And in GATK, there are several options in which I am considering haplotype caller. So haplotype caller is a tool which actually calls the SNPs and all the variations. So for studying, for dealing with all variants like single nucleotide polymorphisms, insertions, deletions, MNPs, that is, and nuclear variants, etc. We can always choose haplotype caller here. Whereas in Freebase tool, there is no such tool. You can directly use Freebase. You can give your reference genome, your alignment result, that is the BAM file, and that's it. So this is a very easy step, but mostly preferred is GATK because it has many customized options. You can also filter your variants after your analysis is done. You can also sort what type of variants you want, etc., in the same tool itself. So the command we use for GATK, as I had said, you give the Java file and the path to your GATK. Then you give your reference genome. In my case, the reference genome is this, which I have copy pasted, a human reference genome. And then I have to give the input BAM file, which was generated in the alignment, and then I'll get an output BCF file, which is in compressed format. And these are the standard commands. You can just either skip it, or you can always or have it on option. You can, when you're practicing, you will always understand. So I can show you an example of BCF file here. This is single nucleotide variant VCF file. So usually it will first contain what is the file format. This is a very important feature of a VCF file. It needs to have the file format is equal to VCF for version 4.2 or any version of VCF file. Otherwise, your system will not recognize that it is a VCF file for further processes. So this is how a VCF file looks. The thing I am not running the commands right now is because that's the reason I had asked you all to finish it yesterday, and I can share after alignment only. You can move forward with variant calling, because this again takes more than 1 day, or sometimes more. Or if you have very high systems, it takes like 6 to 10 hours easily. So this is the file format, that is it is saying that the file is a VCF file, and then it will mention all the filter options we have. So as I said, you can choose if you want to print only a particular type of a variant, etc. Like the allelic depth, and then you can have approximate read depth, you can have the genotype quality, everything. If you want, you can specify. If you don't want, it will take the default values, which is test standard. Even the Fred quality score, it will take the default ones. And these are all the other. This is the header part, right from GATK command line haplotype caller. This is these are all the header part. If you want, you can always read and understand. And then it will just give us all sort of options it has in this, and the descriptions for each of them. And then moving forward, this is where the information of the file starts from. So we can understand, like if it is A, then what it means, allelic count for each alternate allele in the same order. If it is A, this is the allelic frequency. This is AN, that means it is total number of alleles in the genotype which you have called. So you can always refer to the upper header part information, and down from here starts the file information. So these are the contigs in your sample. These are all the contigs in your sample which it was filtered out. And then when you go down, this is where the VCF information, variant calling information, starts from. So you can always check the table heading. So the first column is the chromosome position. Then this is the position of the chromosome. This is the ID of the chromosome. Then this is the reference genome. The reference genome has gene nucleotide in this position in chromosome 1. That's how you read the VCF file. Then you have alternate column. This is your sample nucleotide. So what basically variant calling file will do is, it will take your sample file, and it will compare, and it will map with the coordinates of your aligned bank file, and it will give you a table like this, which mentions your alternative, means your sample's nucleotide, and this is your reference genome file. So basically, to interpret this information, how we go about is this first example only I'm taking. The first variant or alternative nucleotide basically specifies that in our sample we have a T nucleotide, that is thymine, and basically in reference it is G, that means it has changed, that's why it is a variant. And the position is three. This is the position, and in this chromosome, that is chromosome 1, and this is the chromosome ID. You can always use these IDs for further downstreaming analysis and determination of its properties and gene symbols, etc. You can always copy them and paste them in NCBI PubMed IDs, protein DB IDs, and you will get an information if this altered nucleotide is of a gene or a protein, or what role it plays, etc. It will all be there in the other genomic databases. So this is the basic information and how to interpret your VCF file. Then it has given us information about the allelic frequencies, nucleotide, and depth of the allele, etc. So some people, this is minimum quality score and everything. So some people take into consideration the allelic frequency, allelic depth, and etc. Some just give their default values and proceed with variant calling. Anyone has any doubt in this format, or until what I was explaining now? You can always switch your mic. Yeah, no, Miss Priyanka, via Freebase we cannot we do not convert the BAM file. To convert the BAM file from SAM file, there is a tool called SAM tools, and to call the variant from the BAM file, we use Freebase command. That's basically known as calling a variant, not converting. So because when you perform whole genome sequencing analysis, you technically you know that there are certain normal samples, certain test samples or infected samples, and you are expecting variations in that test samples. So to know what variants are present in what samples, and how do you go about it, this is how you call those variants, and how you refer them, understand them, and these are the IDs. You can always take these IDs, and you can always search in your genomic databases. It will give the information each of these ID carries. Uh, yeah. Any queries until now? That is allelic depth, Mr. Arun. In BAM, oh, VCF file you mean? Yeah, it's an allelic depth. Any further doubts? Uh, any doubts? More? Basically DP can also mean filter depth, or the depth of that particular allele as to what you filters you have set. So this is the basic first output of a variant calling pipeline, basically whole genome sequencing analysis. Now we cannot just leave the variants. I mean, of course, if you want only the variants and you don't, you want these IDs, that's a different story. You can just extract these ID column using any method. If you are very good at command line, you can just extract this third column from this file, and you can proceed further and get the information. Otherwise, you can also, what you can do is annotate using different tools. The basic tool we use is snipf and snipset. So snipshift extraction. To install this is very easy. I'll share the link of installation. Hmm, just a second. So there is a GitHub page which will give you snippf snipshift installation, and you just have to click on download snippf. It will download in your directory as a normal compressed file. You just have to extract in whatever directory you want. You have to remember which folder you have downloaded, and then it will have a jar file like this. So the command will be uh, yeah. So this will be the command. Like, this is my location where I have installed and extracted snippet. So I have to give Java jar, and I have to give the entire path. This is called the path of the directory, entire jar file path, and I have to proceed further. So the first thing when we do in snippet is we have to build the database. Technically over here we don't have to, because we are dealing with human genome databases, we don't have to uh, see. So it shows basic command, that is build a database and etc. Or you can also give a command for annotating a variance, and you can also calculate effects. It's the same. Basically, when you give annotate and give your VCF file, for example, over here it will annotate your files, and it will give you the another VCF file which will have a summary of your variants, and what is that variant. Is it an insertion, deletion, or is it a duplication, or is it some other snippet for SNVs, etc. um, and then you have another one which is called snipshift. This is snipshift.jar. Again, you have to use the same command. just, and you have to, it will give you an options over here also. You just have to select which one do you want, if you want to annotate it, or you want to use some other, you want to annotate with multiple entries from DBS and SNPs. Usually what we do is we extract fields from our VCF file. So first we will do snipf, right? So snipf will give me an output again in the VCF format, and then what I can do is I can take that VCF file and I can put it in snipshift and extract all the fields. So it will give me a tab separated file with all individual fields, and it will generate an HTML report. I can show you some sample snipshift file also. Just a second. Um, This is just one example they have given. It's basically an HTML file, and it will just give you a summary of all type of variants, transversions, transitions, and the ratios, missense, nonsense, and the ratios, and SNPs. How many SNPs are identified? How many MNPs have been identified? Insertions, deletions, and everything. So you can always get a summary of your variant files also. So this is basically not a huge step here, but one thing in mind is whenever you're using a new genome, like for example a plant genome which is not saved in snipf. Usually snipf comes with its own list of databases. Like I can run this command for you all and show. Basically, snipf it always has some sort of stored databases. So databases here, I mean, uh, snipf is one such tool which already has the list of all databases and reference genomes in it, and from which database has it. Like for example here, it has all these multiple databases which contain information about only homo sapiens. So it has one UCSC browser's reference genome, it has one the UCSC browsers only known genes reference genome, and then many other databases. So you can take any of them, and you have to just specify the database, and you have to just give your input VCF file and output another VCF file. But in case you have a separate whole new genome, so first thing we can do is we can check in the snipf config file. uh, so this is how it basically looks like, all your snipf config file. So it has information about all the organisms. For example, I'll take Pseudomonas species. So it has a Pseudomonas species genome, which is named as Pseudomonas species genome, and this is the ID. They have copy pasted the reference genomes database. So whenever you are adding a new genome which does not have an information in its configuration file, you also have to follow the same. You just have to, like for example, if you are creating a reference genome for Arabidopsis, of course it will have in this configuration, but just an example, you just have to open this in a text file. You have to add another column. So it will take some time to go in the bottom. Like if you can see, like here they have given mouse here, they have all different versions. So you just have to create one such link name called whatever arabidopsis.genome, and you have to save the reference file as sequences.fe, and you have to just build the database first, add into your configuration file, build the database. First, using the reference genome, then you have to edit the config file. So before you build the database and you have to edit the config file, one important thing is your reference genome file and your annotation file should be in the same directory as your snippf.jar or config files are. Of course, I will share a detailed link. You can understand with examples. I will also give some links here, but I'm just giving a general explanation here. So you have to have all these set together. You just have to build them, and that's it. It'll be done, and you just have to do the same for Snipshift. But Snipshift, you don't have to create all these things because you've already done for Snipf. You just have to remember what is the reference genome you have saved it as, and you have to just give the same as the reference genome, and your HTML file will be ready. I hope you could understand this part. Any doubts, please let me know. You config file. This is the 1 second. I can share the installation site with you. So when you download, sorry, when you download Snipf in your system, only it comes with the config file, and which has all information in it. But if you still think your organism or genome is missing in the config file, you can always add it. So GitHub explains in detail. This is the installation site for Snipf and Snipshift, and I'll also share a simple site which will explain in layman's language. So I have explained it in a very simple way, but only when you practice it will you be able to understand what exactly is happening, in this particular file. So that's the end of whole genome sequencing until the annotation part. There are further steps. Some want to do GWAS. Some applications can be for genome-wide association studies. Some can be QTLC. Then some might want to filter out all the variants and do a phylogenetic analysis also. But these are all different downstreaming analysis. But the first major analysis is whole genome sequencing, and you obtain variants and you understand. You make a list of different variants in your analysis, and it is always a text file. The VCF file is always text file, but when you open it in the text editor, it takes a lot of space. It might, Windows system might get stuck if you don't have enough of space in it. I can show you one normal extracted file. Just a minute. I can show you how Snipf annotates the VCF file and how it prints it. Just a while until you can ask me any queries you have, and you can also try downloading and processing further. Yes, so
ഹലോ ആ വിളിച്ച് പറഞ്ഞു
just a
പറഞ്ഞോ പറഞ്ഞു
second.
ആ ഹലോ ആ പറഞ്ഞോ 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 ആ
So like this is an
ഹോൾ
annotated VCF
ജീനോമിലോ
file. So this is how it will give you a VCF file. With of course it will mention that it is a VCF file. It's of snipf version, and then like here it is giving you what is the what is, again, it's the first column, second, third, until quality is the same as the normal VCF file, but it will just give more information like what exactly does the particular variant do. This is when you annotate the
അതാണ്
file.
പ്രശ്നം.
And so here it has given function
അതാണ്
annotations
പ്രശ്നം
also,
നമ്മുടെ
CDA
നമ്മൾ
length, CDNL, CDS
ലിമിറ്റ്
positions also.
ലിമിറ്റഡ്
So you can
കൊടുക്കുന്നത്.
always change them, and then there are
കഴിഞ്ഞ
particular formats for
ലിമിറ്റ്
gene name
കഴിഞ്ഞു
and gene ID,
അപ്പം എങ്ങനെയാ പ്രശ്നം
etc. So
ആ
it can always change
ആ
the different
റെസ്പോൺസ് എത്രയെന്ന് അറിയില്ല നമ്മൾ
features and
എന്തായാലും
mention
വിൻഡോസ് അല്ലേ
according to
യൂസ്
what
ചെയ്യുന്നത്
you want.
അതാണ്
Some don't want to annotate it, some want to annotate it, and some just want a
അതാണ്
summary of
ആ,
snip.
ഇതാണ്
So
ഇവിടെയും
you just
പ്രശ്നം.
print a snip HTML summary file like
എല്ലാവർക്കും
the one I
ഫോളോ
had
ചെയ്യാൻ
showed you
പറ്റത്തില്ല,
before, and you can
ചിലവരെ
just take
ഉള്ളൂ
a gist of
കോർഡിനേറ്റ്
what all
ചെയ്യാൻ.
variants are present in your file.
അതാണ് പ്രശ്നം.
Uh,
ആണ്, എന്നാലും എല്ലാവർക്കും
I
ഫോളോ
hope
ചെയ്യാൻ
you
പറ്റത്തില്ലല്ലോ,
could
അതാണ്
understand
പ്രശ്നം.
little until now, at
അപ്പോൾ നമുക്ക് നോക്കാം.
least.
നമുക്ക് ഇപ്പം കുറച്ച് പേര് ഓൾ എക്സോം സീക്വൻസിങ് ഡാറ്റ അനാലിസിസ്
I
ചെയ്യാനും
will once again, share the
അപ്പോൾ അതിനൊക്കെ ചെയ്യുന്ന
workflow,
ആളാണെങ്കിൽ നമുക്ക്
and
അതൊന്ന്
you can take
ചെയ്യണമല്ലോ.
any raw data. You can download FASTA files,
നോക്കാം.
FASTQ files, or if you have any rough FASTQ files, please share among yourself. You have to
ചെയ്തെടുക്കാൻ
have two
ക്ലാസ് എടുക്കാൻ
samples, one for normal
ക്ലാസ്
sample
എടുത്ത്
and one should be a
ക്ലാസ്
test
എടുക്കാൻ
sample.
പറ്റുന്ന ഒരാളാണോ എന്ന്
And
നമുക്കറിയണം.
basically that is four FASTQ files. So
അതാണ്
if you
പ്രശ്നം.
can
എല്ലാ
please do
എല്ലാ
that.
ആ ചിലർക്ക്
Then of
വർക്ക്
course they
ചെയ്യാൻ
take a
പറ്റും,
huge
ക്ലാസ്
data
ക്ലാസ്
space,
ചെയ്യാൻ
but
പറ്റത്തില്ല. അതാണ്
that will be
ഇപ്പം
encouraging if you
പ്രശ്നം
could
ഇവിടെ.
practice it.
അവർക്ക്
And I will
വർക്ക്
share all
അറിയായിരിക്കും,
the
പക്ഷെ അത്
logs,
എങ്ങനെ
and you can follow
പഠിപ്പിക്കണമെന്നുള്ളത്
them if you want the
അറിയാതെ
desired
അങ്ങനെ വരും.
result. And
പെട്ടെന്ന്
always email me
ക്ലാസ്സിൽ
your
ആ
query if you want after
പെട്ടെന്ന്
today's
കിട്ടാനും
session.
പാടാ, അതാണ് പ്രശ്നം.
Any
ആ
doubts?
ഇതിനകത്ത് മുന്നോട്ട് പോകാൻ പറ്റുള്ളൂ. അപ്പം നമ്മളത് കൊടുത്തിട്ടുണ്ടല്ലോ. ഉം. ചെ- പറ്റത്തില്ല. അപ്പോൾ നമ്മളത് ഇട്ടിട്ടുണ്ട്. നമുക്ക് റെസ്പോൺസ് നോക്കാം ഇതിൻ്റെ. അതിനുശേഷം നമുക്ക് ഓൾ ജീനോം ഓൾ എക്സാം ഒക്കെ
Does
ചോദിക്കുന്നുണ്ടാവും.
anyone
പക്ഷേ
have any doubts you
കുറച്ച്
want
പേരെ
to
കിട്ടുള്ളായിരിക്കും,
clarify?
അതാണ്. ആളുടെ സംസാരിച്ച് നോക്ക് പ്രിൻസ്. അയാൾ ഫ്രണ്ട്സി കോൺഫിഡൻ്റ് ആണെങ്കിൽ നമുക്ക് സംസാരിക്കാം. അതാണ് കുഴപ്പം. ആ അറിയാവുന്ന ആളാണ്. അപ്പോൾ നമുക്ക് ആ അപ്പോൾ നമുക്ക് ആ ആ
Okay. Of course, I mean, I would ask you all
ആ
to,
ആ
proceed with the alignment and do the variant calling by today and tomorrow.
ആ
I shall be available until tomorrow via WhatsApp. But if you still
ആ
have any queries and if you
കുഴപ്പമില്ല,
still need any
ഞാൻ
help,
നോക്കാം.
you can always contact me
നമുക്ക്
via
പ്രൊസീഡ് ചെയ്യാം.
email. I can
കുഴപ്പമില്ല.
provide my email.
നമുക്ക് മെയിൽ പറയാം. മെയിൽ ആ
Yes, I'll
ആ
share my email ID in the group,
കണ്ടിന്യൂ ചെയ്യാം.
and I'll share a drive link with
സംസാരിക്കാം.
all the logs,
കണ്ടിന്യൂ.
references for Linux. Linux, one thing is most it will be helpful if you could install Bioconda packages. So people who are facing issues with downloading SRA, FastQDump, that is SRA toolkit, I can share one link. So before that, you have to install Conda packages, and it will become easy for your installations. So this will be a link for downloading your SRA tools. You can try with Conda installation, and then you can always try using SRA toolkit, exploring the toolkits also. SAM tools also is of huge. I'll give the list of all the tools like I had mentioned before. You can always, there are multiple tools like instead of Trimmomatic and trimming, you can also use FastP, which is another Conda based tool. Then there is also Galaxy, which is a web based interface where you can also perform variant calling in that web database. The reason why I'm not showing is because I don't want you all to confuse with Linux based and then the Galaxy. But you can also open Galaxy, explore the database, submit your files there, and it will always specify what is the expected input file and what will evolve, what will be the outcome there. And you can interpret based on your uh, choice of study. And don't forget, whenever you install a tool, you can always just so there are always a list of options. You can always take around and check with each and every option it has. You can always go for functions like certain tools annotate, certain tools also do all trimming and mapping at the same time. So you can also do that as well.
Okay, the session is over, Dr. Ishwari.
Yes, doctor. If there are
Okay.
no doubts, I can wind up the session.
Okay, then if any feedback or anything from anyone, you can tell so that we can improve the program. Okay, or else we will wind up the program.
Yes, there's no doubts. You all can also, Linux, Unix is the same. Linux is the operating system, and in Unix is a little maybe different, but it's all the basic is the same.
So
Mr. Arun Jivas, the steps are similar until what I've mentioned, that is variant calling, GATK or Freebase. But after that, you will have to use different packages for Jivas analysis again. So you can read some research papers, and there are multiple tools. You can use R programming also, or there are some Linux-based tools also you can install and run as well. But even that requires a very high computational speed if you are aware.